Okay, so as mentioned, my name is Renad Al-Ghazawi. So I'm going to present our project on differential equation modeling of cell population dynamics in skeletal muscle regeneration using single-cell transcriptomics. And this research was conducted in the Perkins Lab at the University of Ottawa under the mentorship of Dr. Perkins here and Dr. Xiaojun Zhao. And currently, right now, I work as a computational biologist at the University of Texas, specifically in the high throughput core facility. Okay, so in today's talk, I'll just cover the following key areas. So the role of muscle stem cells in regeneration, why they're important, and how they interact with their microenvironment. And I'll talk a little bit about our single cell analysis process and how we use that to extract population data and the development of our ordinary differential equation model and how we calibrated this model to the population data extracted from the single cell transcriptomics and what we gained from this. So let's start. So skeletal muscles have this remarkable ability to repair itself, which is why it takes a few days after intense exercise of muscle soreness. So it just takes 2 to 3 days, or why it requires little to no medical intervention after you pull a muscle. So within skeletal muscles are these things called satellite cells, which are adult stem cells that maintain tissue homeostasis, and they preserve the muscle stem cells' integrity after injury as well. And these cells are found at the periphery of each myofiber, and they're sheltered within a niche microenvironment. So in response to injury, regeneration is orchestrated by a network of interactions with a variety of cell types, including immune cells, fibroadipogenic progenitors, vascular cells, and more. And these interacting cells provide the essential signals that regulate the muscle stem cell's ability to activate, proliferate, differentiate, and do all of these things after muscle damage. So when the muscle is under homeostatic or healthy conditions, satellite cells typically reside in this state called quiescence, which is also a resting state. And this is most commonly marked by the PAX7 marker. So any disruption to this niche, mainly from injury, will take these cells out of their quiescence where they will activate and proliferate. And this is marked by a cascade of MIF5, MyoD, and MyoG expression. They also demonstrate self-renewal capabilities. So once they're activating and proliferating, these cells will start dividing under asymmetric and symmetric division. So one cell will actually just go back and self-renew to replenish the stem cell pool. And the other will become this myoblast, where they will also go along the myogenic lineage and become these myocytes here through differentiation. And then these myocytes will start fusing together to become new regenerated myofibers. So in terms of how the niche guides this process, if we focus on just immune cells, we see this influx of neutrophils, and this initiates the inflammatory response. This is followed by monocytes that transform into M1 macrophages, and M1 macrophages helps the proliferation of activated satellite cells, and also they clear out any debris and necrotic tissue, and then the environment shifts to anti-inflammatory as M2 macrophages predominate. So basically what this does is it will guide the differentiation of myocytes into fusion as well. And then it will also prevent further activation of the satellite cells so that there's no new or more self-renewing cells after sufficient regeneration. So currently there are remarkably very few population dynamic models that focus specifically on muscle stem cells and their microenvironment. And here I highlight some of these studies which employ a variety of approaches, anywhere from differential equation modeling to agent-based modeling. And notably, these models have advanced our understanding of the regenerative process. So as you can see, it explains some behaviors at time points that have not been directly observed in experimental data points. And yeah, unfortunately, they don't account for distinct states such as the quiescent state, activation, self-renewal. And a common practice between all of them is that they combine time course data sets from several different sources due to the lack of data availability. So the reason why that might be a problem, despite the fact that the muscles will regenerate regardless of their injury agent, is because the trajectories can vary due to variable effects of their injury protocols. And this complicates the identification and quantification of specific subpopulations. Also, when combining data sets, there's a risk of simplifying and overgeneralizing unique cell behaviors. Therefore, you would potentially miss out on specific nuances that would be revealed with a unique data set. So by incorporating the use of single-cell RNA, this approach allows for the detailed quantification of diverse cell types, including unique subpopulations of the satellite cells here. So our aim was to develop a model that was capable of simulating post-injury cell type responses of muscle stem cells in their different states, as well as key cell types within their niche using transcriptomics. So I'm sure a lot of you are familiar with the single cell pipeline. So I'll just give you guys a quick overview and then I'll talk about our clustering approach. So we first selected the specific post-injury time course data set, and the initial steps involved quality control and normalization of the data, and then we identified the most informative genes and carried out data correction to mitigate any batch effects, and then we utilized dimensionality reduction techniques to obviously visualize the data. So this was followed by the identification of distinct cell type markers, and we clustered the cells using a combined annotation approach. So what that means is that instead of relying on just a pre-annotated data set to give us preliminary cluster annotations, we also manually identified any non-specific cell types or unannotated clusters by relying on detailed analysis and expert knowledge, which involves splitting or merging clusters based off of their gene expression profiles. So to train our mathematical model, we first analyzed this data set by McKellar and colleagues, and this contained around 90,000 cells and revealing 22 cell types with well-established marker genes. So these markers were first identified by differential gene expression shown on the heat map on the top here. And then this was further validated with established marker genes from the experimental studies in the literature. And that's shown here with this dot plot here. So the step was basically just to ensure that the marker genes selected were not only statistically significant, but also biologically meaningful in terms of the repair process for muscles. And for the purpose of our model, particular attention was given to the muscle stem cells and immune cells. And we subclustered the muscle stem cells shown on the right here, with quiescence on the bottom, activated in the middle, and differentiated on the top and left side. So the same annotation approach was also conducted with the muscle stem cells, and you could kind of see this transition in the dot plot where they go from quiescence all the way to a committed state. And since our goal was to highlight the progressive nature of skeletal muscle regeneration across time, we repeated the single cell analysis using the same protocol with a separate data set. And these graphs here illustrate their immune cell proportions among both data sets. So you can see they show consistent trends except for this peak in neutrophils and monocytes at day 1. This is just a unreported time point in the McKellar study, which is the second data set that we chose. And therefore, we likely missed those earlier dynamics for that specific study. And for the days that they do overlap, so day 0, 2, 5, 7, we see this initial peak and depreciation in their cell type proportions, which is actually really consistent with the different phases of the repair process. So neutrophils first, monocytes, followed by macrophage, M1 macrophages, then M2. And similarly for the muscle stem cells, both data sets mark comparable levels. So you can see most stem cells are initially quiescent, then they shift to an activated state around day 1 to 2, followed by this phase of differentiation by 3 to 5. And these trends also just affirm the consistency of not only the muscle stem cell behavior, but also the stem cell behavior reported by single cell transcriptomic data, as shown by these two independent data sets. So the modeling process began by converting the annotated cell counts for each day post-injury to their proportions, and that was scaled relative to their muscle tissue volume. And just noting here, these clusters on the left are just from the same analysis using different parameters and resolution. I just thought they were prettier, but we repeated this analysis multiple times. So basically, our effort was to incorporate established biological processes and expand on current mathematical assumptions informed by existing models. We then assigned a equation for each cell, estimated the parameters, optimized them using the single cell data, and then the parameters were perturbed iteratively to perform our sensitivity analysis. So, as I said, parameters were first initially estimated based on a confluence of theoretical values and the kinetics of cell recruitment and decay. For example, if you expect to see pro-inflammatory macrophages to exit the system by day 2, we can apply a exponential decay model to roughly estimate its kinetic rate. And by systematically varying parameter values and observing their impact on their model behavior, we identified a set of preliminary parameter estimates, and their ranges were consistent with the empirical data. So then we used solver packages to simulate the model and to observe how well it mirrors or replicates known behaviors over time, followed by setting our error function, which minimizes the difference between the observed and predicted values. So parameters were then refined by applying a quasi-Newton optimization algorithm. And basically this method just iteratively adjusts the parameters to reduce the error function and also improves the model's fit to the data. And then we validated these parameters against a different data set, the McKelly data set, which I showed earlier, to ensure the model's accuracy and robustness. So this is a schematic of our mathematical model. And this was developed from the literature-based understanding of the biological context surrounding muscle stem cells and their niche. And each equation structure is reflective of their processes that's known in the literature. And also that includes anywhere from chemotaxis, phagocytosis, apoptosis, differentiation, proliferation, and more. And these were the 10 cell types that we focused on for our model. So each node here represents a population of one variable, and its change over time is expressed using an ordinary differential equation. And to summarize what's going on here, basically the process begins with muscle injury, and that's represented as damaged myonuclei. And this basically leads to recruitment of inflammatory cells, so neutrophils, and that helps clear the damage and release signals to attract monocytes. And then the monocytes basically transform into M1 macrophages. So these M1 macrophages peak starting around day 2, and then they clear out any debris, and then they switch to M2 macrophages, which promote inflammation resolution. Then the muscle stem cells are basically activated from their quiescent state as well, simultaneously. And
sorry, you're good, keep going.
Yeah. And they eventually fuse together to become new myofibers. So I'll also note that the model also uniquely incorporates the regulatory role of M2 macrophages on satellite cell fate, which was actually not considered in previous models. So I'll examine one cell type in more detail for you guys, and I'll very briefly explain the others. So what you're looking at here are the trajectories obtained from the simulated model trained on McKellar's dataset. And the second data points, so the red ones, are basically the separate single cell dataset. And these trajectories are simulated to reflect the repair process from 0 to 7 days post-injury and are represented in cells per cubic millimeter of muscle tissue volume. So focusing, as I said, on the quiescent satellite cells and its observed behaviors. So it captures how well neutrophils respond, or how well quiescent satellite cells respond to neutrophils and damaged myonuclei, as well as their self-renewal, which is influenced by M2 macrophages. And then the last term basically just represents a feedback loop, ensuring quiescent satellite cells, their levels adjust to maintain homeostasis post-repair. And if we look at the right, quiescent satellite cells basically demonstrate heightened sensitivity to activation signals from damaged myonuclei. And this also may suggest damage severity may significantly drive the rate at which they exit the cell cycle for differentiation. And across all three states, parameters involved in the inflammatory process or response have a higher impact. So with activated satellite cells most affected around day 2 to 4, and myocytes around day 3 to 5, as I said, was also reflective of their roles in muscle repair. And overall, you can see that the model mirrors general trends in immune cell responses. So if you take a closer look at the neutrophils and monocytes, those are the earlier cell types that are influxed upon muscle injury. And it suggests that some of these immune cells may peak sooner and at higher levels than reported data shows, suggesting that the acquisition of these cells or samples were not initially captured at those earlier times. So those could be potentially revealed by this model. And the model's predictions for myonuclei, although not directly measurable by single-cell RNA, indicate a timely clearance by day 2-ish. And this is also consistent with observed muscle repair timelines and the rate of, activated satellite cell proliferation appears to be a significant factor, with the model showing heightened proliferation rates contribute positively to the regeneration process throughout the regeneration, all throughout day 7. So to summarize, our model incorporated 10 cell types, 22 parameters, introducing interactions that were previously unmodeled, yet hypothesized in existing literature, such as the influence of M2 macrophages on the dynamics of activated satellite cells. And a key distinction of this work is this approach to data utilization, which also differs from conventional studies, which typically pull data from multiple different sources for model fittings. And it also potentially uncovers dynamics at specific time points that are not recorded in experimental settings. So this may enhance our understanding of the underlying biological processes. And lastly, sensitivity analysis has highlighted some parameters of high sensitivity, pointing to avenues we should probably look into for future research. So while our model is a step forward in simulating distinctive cell states, we suggest that future iterations could benefit from adding different cell types like fibroadipogenic progenitors, vascular cells, and others. And as research advances, particularly in dystrophic muscle studies, there's a potential to use this model as framework to contrast cell populations between diseased and healthy tissues. And on that note, I'd like to acknowledge everybody in the Perkins lab, especially Dr. Perkins and Dr. Shao's mentorship throughout my graduate studies. And I hope you guys learned something from this talk today. So thank you.
Questions? Go ahead.
Next one. From the single cell analysis, you have the transcriptomic state of the cells. Have you thought about including the gene regulation into your model? And can follow the transcriptomic state you see in single cell analysis?
Could you repeat the question for the virtual audience?
Sorry, what I think you're asking is something like a pseudotime trajectory or finding its transcriptomic states along.
Right, to also include the genetic regulation into the dynamic model.
So we did consider it, so this model was basically just like a pilot study just to see if it was able to simulate the post-repair timelines adequately with the data that we did have. Unfortunately, we didn't have that much available data, so for future studies, we might definitely include that for sure.
Any other questions? So I have a quick question. The parameters that you learn from the data, you assess that they generalize to the test data using the other source of the other data set that you were describing. Do you have a sense of whether the parameter values themselves are kind of identifiable and unique, or are there multiple optimal fits?
That's a good question, actually. So there was actually multiple optimal fits. So through our optimization, we've repeatedly have done it multiple times, and there's a range of parameter values that are adequate for simulating all 10 cell types, but because it's a really big system, so 10 cell types is kind of hard to optimize for all of those 22 parameters. We selected this range that was somewhat adequate for initial estimates. Yeah, so I don't think there's like one value that's what we can say is like this is the value of or the kinetic rate that we're set on. I think there's a range that we can play around with. So yeah.
But in particular, are there like a pair of cell types whose proportions are correlated so that you
Oh,
will
yeah.
get confused whether it's this or that which is more important?
Definitely, they are like they are all proportional to each other. So there's direct transitions from one cell type to another. So their parameters are directly correlated. Yeah.